These are the recorded lectures for Chapter 28 on invertebrates. These are our animals without a backbone. Make sure that you know the characteristics that all animals share. So if an organism is classified in kingdom Animalia, then it is multicellular. It's a heterotroph, meaning it consumes its food by ingestion. And then a comparison of fungi and animals. Fungi digest their food externally and absorb the nutrients, while animals ingest or eat their food, and then the digestion and absorption is internal. And here we see in picture A, we see the nervous tissue, neurons and nerve cells of animals. Of course, here we see, is that a falcon? We know it's a bird that's a predator that's eating its prey. And then we have a cheetah, fastest animal on earth. And then we have a ball of cells called a blastula, which forms into the embryo of the animal. I guess showing that animals are multicellular, and here we've got animals eating by ingestion, and then animals can move. Now, one thing you should know about movement is that all animals can move at some stage of development. There are some animals that are sessile, some aquatic animals like sponges and sea anemones that are sessile. It is believed, based on evidence, that choanoflagellates, which are protozoans, resemble the single-celled ancestor of animals and fungi. All right, one thing that we talk about with the different animal groups is their body symmetry. Symmetry is a pattern of similarity observed in objects. So even non-living things can have symmetry. But there are three types of symmetry that we see among animals. Some animals are asymmetrical. The sponges are asymmetrical. They don't have any particular body shape or body symmetry. Then we have animals that have radial symmetry. So you can make a longitudinal cut through the entire animal in several different directions and get two identical halves. So a jellyfish has radial symmetry. Then other animals, you can only make a longitudinal cut in one direction to get two equal halves, and that's called bilateral symmetry. If an animal has bilateral symmetry, then it's got definite right and left halves. These animals also tend to have a trait called cephalization, which is the development of a head region with a brain. The first tissue layers are called germ layers, and they give rise to organs and organ systems. Animals that have only two tissue layers when they are developing in the embryonic stage, they're called diploblastic. And animals that have three tissue layers are called triploblastic. Among the triploblastic animals, protostomes are the ones whose first opening becomes their mouth, and deuterostomes are the ones whose second opening becomes the mouth. So in protostomes, the first opening is the mouth, and the second opening is the anus. And deuterostomes, the first opening is the anus, and the second opening is the mouth. So here you can see radial symmetry, and this looks like a sea anemone or a hydra with a polyp body form. So it's related to a jellyfish. And you can make, if you look at these glass dividers, you can make cuts in any of these directions. You see three different glass dividers, and in any of those dividers would produce equal halves. Whereas with the turtle or tortoise that we see here, I'm not sure which one it is, we can only make one cut through that animal to get equal halves. And you can see that the turtle has a head region, so it does have cephalization. and it has a definite left and right side. Oh, these are terms you might find useful. The dorsal region is the back of the animal, and the ventral is the stomach region of the animal. Anterior means toward the head, and posterior means toward the tail. When we use the term cleavage, this is what happens with the zygote. Immediately after the zygote forms, it begins to divide, but when it divides, the cells that form don't make the overall group of cells is the same exact size as the zygote itself. So the zygote divides into two cells, and those two cells are the same size as the original zygote. So it's cell division without growth. The cells just divide and divide and divide into a ball of cells. For protostomes, the cleavage is spiral, and for deuterostomes, it is radial. Then the ball of cells that forms is called the blastula. In protostomes, the first opening of the blastula becomes the mouth. In deuterostomes, the first opening becomes the anus. The second opening becomes the mouth. And some protostomes, and all deuterostomes, have a body cavity called a coelom. In protostomes, the coelom forms by splitting of the mesoderm tissue, and in deuterostomes, the coelom forms by an outpocketing of the primitive gut. So let's look at some pictures so that you can see what I'm talking about, because that was a lot of information. So here you can see in protostomes the cleavage of the blastula, which is the ball of cells that forms early in development. Cleavage is spiral and determinate. And then in deuterostomes, the cleavage is radial and indeterminate. I doubt if you'll have a question about that, but if you do, I don't go into a lot of detail because it's really not that important for our course. That you understand the cleavage, you understand what this means really, but that there is a difference in how protostomes and deuterostomes, how their cleavage occurs, which is when the blastula forms early in development. Then you can see in the next set of images where it says fate of the blastopore. The blastopore is the first opening that forms in the blastula. Okay, so you can also see the three tissue layers. On the inside, the kind of tan-colored tissue is called endoderm. The blue tissue is called ectoderm, and the pink tissue is called mesoderm. So you can see that the ball of cells is going to kind of form an indention or an invagination here. When it does form that indention, that is the blastopore, which becomes the mouth in a protostome. In a deuterostome, that first opening, that blastopore, becomes the anus. And then you can see how the mesoderm forms, how the coelom forms. Okay, so inside here in this space, which is inside of the endoderm tissue, that's going to be the gut. That will actually start at the mouth and end at the anus. And that will be true no matter if it's a protostome or a deuterostome. Inside here, this space is the gut. But the body cavity, the coelom, is the space that's inside of this mesoderm. Okay, so if you notice, the mesoderm tissue forms differently in protostomes. And the coelom forms by a splitting of the mesoderm, whereas the coelom forms by an outpocketing of the gut. See how the mesoderm kind of makes an outpocketing early on, and then the mesoderm tissue forms, and then inside the mesoderm is your coelom. So the coelom is forming by an outpocketing of the primitive gut. So anyway, those are differences between protostomes and deuterostomes. For us, really, you just need to know if it's a protostome or a deuterostome. And the most important difference is that protostomes, the mouth develops first. Deuterostomes, the anus develops first. Now, our first phylum is the phylum Porifera, and the only animal in that phylum is the sponge. So these are our simplest animals. They don't even have true tissues, so they lack the tissue level of organization. And they only have a few cell types. They have sac-like bodies that contain many pores, which makes them sponges. They are, as far as the way that they feed, they are sessile filter feeders, so they continuously feed, and they can reproduce asexually by fragmentation or budding, and they can also undergo sexual reproduction. In that case, they'll produce a flagellated larva that can swim to a new location, and then it will grow into an adult that is sessile. So again, animals move at some stage in development. So even though sponges, adult sponges, do not move, the larvae do. They do swim. And here you can see the sponge. What happens is the way that they filter feed is that water moves into the sponge through its pores, and then it passes by the flagella on these cells called collar cells. And those flagella help sweep the particles of food into the cells of the sponge. Then the water will flow out this opening called the osculum. You can see the cell types. The collar cell or choanocyte is the one that's similar to the choanoflagellates, the protozoans that are thought to be most closely related to the animal ancestor. And then we have some epidermal cells. We have an amoebocyte, which is where the digestion occurs, and spicules, which are kind of give the sponge like a skeleton. Then we have the phylum Ctenophora. The C is silent. The phylum Ctenophora are comb jellies, which are related to true jellyfish, but they're not because they don't have stinging cells. So comb jellies are small, transparent, and sometimes bioluminescent, meaning they can produce their own light. If you have ever been to the aquarium near Atlantic Beach in North Carolina, that aquarium has a tank of comb jellies, and it's in a corner, and they kind of give off this blue light, and they're really cool to look at. The body of the comb jelly is composed of a jelly-like substance called mesoglea, and it's the entire body that's composed of mesoglea, whereas in jellyfish, only a part is mesoglea. So phylum Cnidaria are similar to the Ctenophora, but they have specialized stinging cells called cnidocytes. And then inside is a fluid-filled capsule called a nematocyst, and they sting their prey. And they have a two-layered body sac. The inner layer is called the gastrovascular cavity because it serves as gastro meaning stomach, so it serves as the digestive system, and vascular meaning circulatory system. So it's kind of a combination digestive and circulatory system, which is not very efficient, but it works just fine for the cnidarians. Now, we know jellyfish have a certain body form, but sea anemones have a kind of a different body form. So the jellyfish body form is the medusa, and that is where the mouth is directed downward and the tentacles are directed downward. But the sea anemone's body form is polyp, and that's where the mouth and the tentacles are directed upward. Here's a picture of a typical jellyfish, medusa body form, and then picture A on the left is showing you a Ctenophora. This looks just like the ones in the aquarium that I was telling you about. There's just a tank full of, it looks like almost, well, maybe even hundreds of these in the tank, and they glow, they're bioluminescent, but they don't have stinging cells. So there are lots of different types of cnidarians. Sea anemones are sessile polyps. You probably remember the sea anemone from the cartoon Finding Nemo. That's where a lot of us learned about sea anemones and clownfish. But they are sessile polyps, and corals are similar to sea anemones, but they're encased in a calcium carbonate shell. Once the animal dies, they leave that behind, and their shells are used to build the coral reefs. So they form the Great Barrier Reef along the coast of Australia and other coral reefs in the tropics. Okay, so near the equator. So here we see pictures of a sea anemone, a cup coral. It's rare that we ever see the animal that lives inside the coral. And here we see a Portuguese man-of-war, which is actually a colony of individuals rather than one single animal. Then we have a jellyfish. Another type of cnidarian is a hydra. These are found in fresh water, ponds and lakes. They have the polyp body form. The true jellyfish has the medusa body form. Here's an anatomy of a hydra, and then this is a cool picture. Picture B is showing you how hydra can reproduce asexually by budding. So the little baby will grow on the side of its parent, and then it's just a clone of the parent because this is asexual. And then it will break off and grow into an adult hydra. Now, the spiralians are animals that include the Lophotrochozoa, which are divided into two groups, but honestly, we are not going to spend a whole lot of time with the Lophophorans. The bryozoans, brachiopods, phoronids, they're not as well known, but you will recognize these are not protozoans. What is this? This is a mistake in the notes. Let's fix it. I'm going to fix it right now. So this should say protozoans are not even animals, right? They're in the kingdom Protista. So this should say trochozoans. Okay, trochozoans. And the trochozoans are the flatworms, like tapeworms, the rotifers, which are microscopic, but they're very cool to look at, mollusk, which would be like your oysters and clams and the octopus and snails. These are all mollusks. They have a soft body. The annelids, such as the earthworms, these are all in the group of animals we call trochozoans, and they're named for their larvae. They have larvae called trochophore larvae, but they all have bilateral symmetry. So there's only one way that you can cut them in half and give you two equal symmetrical halves. They all have 3 germ layers, so they are protostomes. That means that their first opening is the mouth. Some have a true coelom and some don't. So we'll start with the phylum Mollusca. And actually, I just realized this has gone 18 minutes. So we're gonna start with the mollusca in the next recording, and this is slide 24.